So we'll just do exposure, high-level exposure to various peptides from a recombinant peptide, just to see if we can get anything approximating the symptoms that have been reported, either from virus or from vaccine. There's a number of, why are we trying to do this? Well, there's concern that there are, how should we say, epitopes that have been packed into the genome of SARS that stand out. So, what are we aiming for? So, we want to try to understand what's the impact of exposure, and some people are going to plot towards vaccines that have done it, and some will say, or I would say you can't dismiss the importance of viral impact as well in the emergent states and disease burden that is that we're seeing right now. And why would you get this excess disease burden? And one of the hypothesis that I would put forward, and have been for a long time, is that it's exposure to epitopes of concern that cause an increase in inflammatory tone, coupled with immune dysregulation, that lead to, essentially, dependent upon the individual, a whole suite of medical issues. They could be mild to severe to lethal. And in the bigger picture, I think that these was very likely the opening salvo of perhaps an escalating war that I don't think we've seen the back of, despite Moshiach Ben Trump coming down and promising peace and joy for everyone. I remain to be convinced of that. And so, what are we testing on? Well, hamsters have, fortuitously, Syrian golden hamsters have a ACE2 receptor profile that's very, very close to the human, and the Syrian golden hamster has become a sort of useful model in the study of SARS. Now, I think there might be another reason why the hamster is important, and that relates to these amyloidogenic epitopes. So, who has a, how should we say, well-developed colony and breeding program specifically geared towards prion biology? And that's Rocky Mountain Laboratories. And I think we should be taking a very close look at the output that's come from that particular institute, and how it could have been leveraged to achieve strategic goals. And so that's where we're aiming at the moment, to try to answer some of these questions. And so we were in the process of sort of moving. You've got parallel tracks of experimentation. So vaccine exposure to, we can't work directly with virus, so we choose the peptides of concern, and we expose that way, or we will expose that way, and then we try to understand or see if we can get anything that we can work with. And if we can get something that's tangible and repeatable, we can perhaps work towards testing mitigation strategies in this instance. The hamsters are there. That's all we can do at this point. And so, the hamsters that had vaccine exposure, they look fine, right? Even though I stuck it right in the heart, just, and 100 times the dose that you would give normally. Now, there's many reasons why they appear normal, and we're not in a position to be able to do the histology. So that's another thing that I wanted to bring up, is just, it takes time, and you've got to sync things up such that you like, so in using these peptides, right? So 100 micrograms of spike protein, for example. That's a lot of that's significant money. And I want to say, what was that? I want to say it was $1,200-$1,300 for 100 micrograms. And um, when you've got a limited, like once you've put it into solution, you've got a limited time window in which you can use it. And so we've been waiting for a piece of equipment called a microtome. That's the paperwork is done. We broke the back of that last week, and I'm just waiting on it arriving now at the lab door. And and so it was at that point where things start to come together, and you say, okay, right, we now buy Prion protein, and we begin the testing that we can do around that, which is why it's taken to this time where to be able to use the RT-QuIC, even though the machine has been installed since I would say it was the late July, and now, like I say, things are sort of thinking together, and of course there's would you say I want to say interference, but like when you're it's a kind of metaphysics of laboratory work, and I've always maintained that when doing an experiment, it's impossible to contain it entirely within laboratory, and when you're pushing against the sort of fabric of nature and reality, it tends to push back in unexpected ways. And this now brings us up to date with, or close to up to date with, like, are we making progress in the lab? So myself and Brad have had to basically do RT-QuIC. And so, what do we, so yeah, that's picture of RT-QuIC machine, our workspace. This is me starting to get ready, getting out equipment that we need. Oh, fucking Chrome. Yeah, well, load it again. That work. Weird. See, I don't know what's wrong. Well, I've got my suspicions. But anyway, me doing more stuff. Me doing more. Let's see. That's on the day. Experiment. And this is me basically preparing peptides, getting them into a sort of workable format and bulk solution. And let's see, that's the plate being loaded. And the plate as it goes into the machine. And like I said, I would have wanted to show this live. We video recorded some. Let me find. So it's not that, is it? This. 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 Yeah, this. I think this. Oh, okay, I hope so. Right. Just show me that. Says what? 1:46, Sunday, November 10th. AM. We're setting up for RT-QuIC run 1. So just for some context, this is us getting ready, and I just want to, as a record of, when you do experiments these days, you should probably be videoing as much as possible. A good micropipette is a joy to use. Well, I wish we had good micropipettes, and one of the, how should we say, look, it's not until you do the experiment that you find where it can be improved, and one of the things that we need to improve is we need more wide range of pipettes that can do different sizes, and I'll make sure that when we run next time we have that. And there was just, I didn't have, although we've got a whole bunch of glassware and stuff, I would have liked more, I don't know, just more kit available to me to do. And so what are we doing here? I'm basically just, I wanted to just show the time that we were doing it, and just what we're working with, and that's basically what you're going to listen to now.
Brad, just to witness this as well.
What's that?
Sealed 5-flavin serial number T351625G. Confirm that's sealed. Get some gloves.
So yeah, science can be a little bit boring, but I want you to see that it was real. We did this, right?
Alright.
Again?
Yeah, do you want to just confirm that it says that?
T3516. That one?
Yeah, 25G, and it is. Just give it a.
Very sealed.
Oh, you got your phone just to fucking. Ah, we've got that. It's alright, it's alright. Right. Peptides. Recombinant SARS nucleocapsid protein. Yes.
Yes.
Human coronavirus SARS-CoV-2 envelope protein peptide.
Yes, confirmed.
Lipopolysaccharide from E. coli, LPS.
Yes, confirmed.
Neutrophil elastase, human.
Confirmed.
SARS-CoV-2 spike protein, full length.
Yep, confirmed. They put down there. You can see it there. No, just press up. I think it's just like there.
Safety cabinet.
Speed safety.
Yeah, but kind of just put in. Stop.
Put your arm off.
Yeah. SARS-CoV-2 spike S1 subunit.
Yep, confirmed.
Human prion protein.
Confirmed. Why can't we just have your birthday on?
Well, you want to be happy when you're stepping into where we're just about to step. You might as well have a smile and a grin on your face.
Did I have COVID vaccines? No, I didn't. If that's of use to you, but no vaccine for me. I think I had SARS early. I think I got it in the super spreader event that was Korea. Yeah, a very, very unpleasant, and yeah, I didn't think I was going to make that one, but I did. And
Right. Human PrP protein.
Confirmed.
Alright. What was the wait for the
I don't know. And look, there's basically hours and hours and hours of recordings. So yeah, that's me prepping the stuff. You've seen the pictures for that. Let's see.
Anything else? You come to this, there's thousands of objects that defy estimation.
But yeah, if you would like to, we need more pipettes. Send shekels, folks. None of this is cheap. You just looked at. I want to say that's over $10,000 worth of recombinant peptides that I just put into solution. Nah, just messing around with right here. That's me diluting down everything. So that got done, and I tried to the handheld cameras ran out of memory running to the running through the process. So the only what I did find where did I put that? I tried to find the time from the lab surveillance cameras where we were doing RT-QuIC, and I couldn't. The closest I could find was where was that? Here, and I want to say, yeah, it's this. No, not that. Yeah. So I don't know. Just explain what you're looking at there. So there's the RT-QuIC machine here, right? So there's a laptop. You can see like a map that we've printed out for the wells, obviously the workspace where I was working, and what you can see there is, and I haven't processed the data yet, but I'll tell you what I know and I could see as we watched the reaction take place. So there's no there's no Paggy hotline support that you can call for trying to do these experiments, right? So you have to try to work out the protocol that you're going to use. You've got to program it into the machine, and then you try to watch the or I was watching the raw data, and basically what are we looking for is the thioflavin, which we were talking about, saying, look, that's sealed, and you can see me. If people want evidence, you can see me weighing it out and putting it into solution, etc. And that thioflavin will fluoresce if there is amyloid fibrils forming in peptide structures. Why does it do that? Well, for whatever molecular magic and flukes of nature there are, these were they benzodiazoles, I think they're called. It's the type of molecules as a classifier, but basically they intercalate between the fibril or the oligomers as they're forming and sticking together, and what that does is open them up so that when they get hit by fluorescence light, they'll emit a photon. And with the raw data off the first run, and this is basically the result that came out as of today, the we didn't see anything. Now, again, this is the first time that we've used the machine. first time running through the experiment protocol, etc. And we'll try again later today to repeat the experiment and just to work out why. Well, like I say, I haven't extracted out the data fully to examine it, but my impression of just looking at the numbers coming in was there isn't a reaction happening. And if you're paying attention to what the list of peptides that we were naming as I was showing the video, one of those was lipopolysaccharide from E. coli. Now, why would we have lipopolysaccharide from E. coli? So the issue you have with something like RT-QuIC, where essentially you're looking for you're trying to induce the chemical conversion of the peptide into the scrapie or amyloidogenic form. You want some way of having a positive reaction, because in a sense, when working with the SARS peptides, we're not sure if it'll trigger RT-QuIC, the RT-QuIC reaction. The theory and the informatics says it should, but we don't know. We don't know that for certain, and again, there is published work out there which says it is forming amyloidogenic fibrils, but what to trust and what not to trust. And so lipopolysaccharide, endotoxin, is it's been known for quite some time that it can induce the conversion of prion protein to the scrapie form. And I thought, I don't know, what time is it? 3:55. Did the control work? No, not as far as we can tell. So, and again, there's a learning curve with this process, right? And so, we got no signal across any of the wells that we analyzed or set up and prepped. Now, again, where did the failure mode occur? There's a couple of places where I think it could have done, but the thing to do is just to keep repeating and working through the steps so that you're confident that it's you're doing everything that you can to induce the reaction as has been reported in the scientific literature. And so if I had so I thought about maybe reading this paper as a sort of grounding as to why use endotoxin. Start again in a few hours, and I want to get it right, and so I want to be confident that I'm in the best condition to be able to say that the data was as clean as we could get it. And so I think maybe what I'll do is I'll put the link, and I'll read out the abstract, and then hopefully it makes sense. People that sort of go back and try and watch the video, you understand why the endotoxin step is in there, because working with the COVID or the SARS peptides, it's an unknown whether the reaction happens. Whereas in the literature, it says that the reaction between prion protein and endotoxin is a surefire way to induce the scrapie form. So, did the control glow? No. And so it didn't. And so I'll just put the link for this paper. I'll read the abstract and then I'll think about wrapping this up. All right, so lipopolysaccharide-induced conversion of recombinant prion protein. Hello, something's beeping at me. Let's see. Just get the control to work before you use more on vaccines, hamsters, blood tissue. Yes, that's the whole point in doing this, right? You must have a reproducible model before you go off and start making claims about anything. And I don't see a whole lot of that type of rigor being brought to bear, I'm afraid. And the problem is, though, is that it takes time to do it, and it takes money and time and resources, and there's there are limits to what we can do, right? You have to accept that, right? But this is about as close to, I don't know, there's a few other bits and pieces we could add, but like, can we induce the amyloid conversion? And then if we can induce it at just but what's basically a stoichiometric chemical reaction, then we can feel confident that we're doing something in the animal model. Because right now, what does our data tell us? Well, if you try to stick it in the heart, in the method that we used at the time, we couldn't tell a difference between control or vaccine. Both showed reductions in behavior, but you try getting a big monster to stick a steel cannula in your chest and see if you feel like running in the exercise wheel the next day. Anyway, let's just go through that abstract of this, and then I'll think about wrapping it up. So the conformational conversion of the cellular prion protein, PrP C, to the beta-rich infectious isoform PrP scrapie is considered a critical and central feature in prion pathology. Although prion protein scrapie is the critical component of the infectious agent, as proposed in the protein-only prion hypothesis, cellular components have been identified as important cofactors in triggering and enhancing the conversion of PrP C to proteinase K resistant PrP scrapie. Now, why is this important? Because what is lipopolysaccharide? Well, it's what it says. It's a fatty sugar complex that is essentially on the outer membranes of, I want to say it's outer membrane of E. coli bacteria, and I want to say all E. coli bacteria are sources of endotoxin, as are many other bacteria as well. But E. coli in particular is standout. It's a model organism for production of endotoxin, and the and so when dealing with disease burden at a large population scale, generally you have a spectrum of how how exposure to pathogens will present itself, and it's quite possible that people who have what could be described as quote unquote dysbiosis are in a primed state that allows exposure to amyloid inducing peptides to wreak far more havoc than were you to be in robust and full health. And there's questions around why are these peptides, these amyloidogenic peptides, why do they have this property? and why are they correlated? And I think um, well, I don't know, maybe I'm showing my biases, but I think the weight of evidence to them being causal to disease states tilts very strongly in that direction. But then why some people and not others? And you're always looking for what could be contributory factors, and a big one would be and you can just think about gut health in this instance. What causes the individuals to be or some to be more susceptible than others? And this idea of the gut-brain axis and the biome and also virome state is a critical one. Like the orthodox science is in that space very strongly now. What tended to be the I want to say like how to describe. So people who were very health conscious, or people who had contact with disease, particularly chronic conditions, they tended to think more, or they're more flexible about those types of contributory factors to disease state, and people, and as they've tried to navigate the disease condition themselves, have they realize the importance, and eventually, if there's something there, then you hope that the the science that can bring actual, like, essentially infinite resources to bear does eventually figure it out and work towards and try to understand it. And this interplay between just what's if I'm not a gut specialist, right? So I don't know like the full spectrum of what should be a healthy gut biome. And this is that's other people's area of expertise. But the idea and principles around a healthy gut and is a foundation on which to build a somewhat healthy mind and a brain, and hopefully through that you head and towards a healthy mind and spirit. And so lipopolysaccharides, endotoxin, etc., is a very, if it works in this lab environment, is a very tempting experimental approach to take, because if it works and it acts as a good positive control in our reaction process, in our scientific investigation, then it's a very no, convenient is the wrong word, but it's a sort of low hanging fruit that you can aim at, in which there are ways to mitigate any potential harms that come from exposure in this instance, amyloid exposure, prion exposure. So that's why we would use lipopolysaccharide, and why you should pay attention to this particular result, even though it can seem somewhat cryptic if you're not paying attention. So let's see, a number of in vitro systems using various chemical and or physical agents such as guanidine hydrochloride, urea, SDS, high temperature and low pH have been developed that cause PrP C conversion, their amplification and amyloid fibril formation are often under non-physiological conditions. And that was another tempting reason to use the endotoxin approach, because you don't have to put in, you can keep the medium, the buffer medium the same, the reaction temperature the same, and you can make them as what would be physiologically reasonable, right? So that's another reason as to why it's worth pursuing and putting effort into pursuing the particular line of investigation. So these authors say, in our ongoing efforts to look for endogenous and exogenous chemical mediators that might initiate, influence, or result in the natural conversion of PrP C to the scrapie form, we discovered that lipopolysaccharide, a component of gram-negative bacterial membranes, interacts with recombinant prion protein and induces conversion to an isoform richer in beta sheet at near physiological conditions, as long as the LPS concentration remains above the critical micelle concentration. More significant was the LPS-mediated conversion that was observed even at submolar ratios of LPS to recombinant prion protein. I'll read this paper because it's an interesting one, right? Because it sort of helps broaden the scope in which you should be thinking about how these agents are interacting with each other to cause not just acute but long-term chronic conditions. And you're going to fix the world by fixing those. No, not all of it. Are there other problems to fix? Yeah, maybe. But the little lab that we've managed to put together, right? And that's just a little bit of the lab, right? There's other parts that hopefully will get finished off this week, if we're lucky, and we can start doing the more, what to me are more interesting studies. But then, if we don't see anything with our RT-QuIC approach, should we? There are ethical considerations to think about in this instance. So, I think I'll stop there.